Transfer the MEA plate with the inner box into the clean bench. Remove the MEA plate from the inner bag and rinse each well surface with sterilized distilled water. Repeat the rinsing process 3 times. Pour 70% ethanol into each well for sanitization and leave the MEA plate for 15 minutes. Remove 70% ethanol from each well after 15 minutes. Rinse each well surface with SDW 3 times. Rinse the surface of the top lid and the cell spotter 3 times as well. Dry out the MEA plate, the top lid and the cell spotter completely under UV radiation for 15 to 30 minutes. In subsequent processes, handle the MEA plate after drying by putting it in a sterilized container. Pour 200 microliters of 0.005% PEA solution into each well. Incubate the MEA plate filled with 0.005% PEA solution under 5% CO2 at 37 degrees Celsius for 10 minutes. Remove the PEA solution from each well and rinse with SDW at least over 4 times. Dry out the MEA plate, the upper lid, and the cell spotter in the clean bench overnight. In this video, commercially available frozen cortex neurons will be seeded and cultured. Pour 200 microliters of 2.5 micrograms per milliliter I matrix 511 solution into each well. Incubate the MEA plate filled with I matrix 511 solution under 5% CO2 at 37 degrees Celsius over 60 minutes. Prior to thawing neurons, warm culture medium containing 5% FBS to 37 degrees Celsius. Remove the frozen neurons vial from the liquid nitrogen container. And thaw the neurons at 37 degrees Celsius for 150 seconds. Gently transfer the thawed cell suspension into a 50 ml centrifuge tube. To collect remaining neurons in the vial completely, pour 1 milliliter of warmed culture medium into it. And then collect all the remaining together with the 1 milliliter of medium. To avoid osmotic shock, gently and slowly add the medium into the 50 milliliter centrifuge tube at a drop per 4 to 5 seconds. Add another 3 milliliters of medium to make the total volume 5 milliliters. Add the first 1 milliliter over 30 to 60 seconds and the remaining 2 milliliters over 30 seconds. Gently mix the suspension and medium by inverting the tube 2 times. Count the number of viable cells. After counting viable cells, adjust the concentration to achieve your intended concentration. Transfer sterilized cloning rings into sterilized dish. Stand the rings vertically and shake the dish to check for defects. Wet the inner surface of the ring with a small amount, 5 to 10 microliters, of medium. Transfer the MEA plate treated with iMatrix 511 from the incubator to the clean bench. And mount Med Cell Spotter onto it. Remove iMatrix 511 from 4 wells. Quickly insert a cloning ring into each 4 well. Drop cell suspension into each 4 ring. Repeat these steps every 4 wells to prevent evaporation of the cell suspension. Add 200 microliters of culture medium from the dedicated groove for it. To prevent evaporation of medium during incubation, place small dishes filled with SDW in the container. Incubate the MEA plate seeded with neurons under 5% CO2 at 37 degrees Celsius for at least 60 minutes. Remove all cloning rings from the MEA plate. Remove the med cell spotter from the MEA plate. Add 300 microliters of culture medium to make total volume 500 microliters. Replace the culture medium completely into new medium pre-warmed to 37 degrees Celsius on day 1. Thereafter, replace half of the medium every 3 to 4 days. Replace the culture medium completely again on the day before data acquisition, day 18. This scene shows the full replacement on day 1. The dedicated medium containing 5% FBS for the commercially available neurons is used for cell seeding. And it is fully replaced into brain fizz for culture after day 1. Place the MEA plate with cultured neurons on MED64 Presto amplifier.
思いっきり画面に映ってますけど、先撮ってたやつ。
Wipe any dirt or water droplets on the terminals of the MEA plate before covering. Synchronized bursts can be observed in primary neurons at least on day 19, depending on the culture condition. For drug treatment, remove the cover and administer the drug by pipetting. In this scene, 10 microliters of medium is extracted first. Next, 10 microliters of the drug solution is added into the upper area of the well and agitated. And then 10 microliters is aspirated to add to the lower of the well to achieve the intended final concentration.